Here we go with chapter 11, which I really do love, but I'll tell you, this opening vignette and picture are some of the most disturbing things I've ever seen, and I really don't understand why the text did it. So there's a discussion of that disgusting first page, and it's worth looking at. Boy, I had never seen that before, to tell you the truth. So narcotic analgesics, I would say opiate analgesics. The book says opioids, but in this chapter, it will slip in a few points and use the term opiate. This distinction between analgesic and anesthetic is worth thinking about. Now, an means without, and algesia is pain. So analgesia means without pain. Aspirin is an analgesic. It really doesn't remove pain typically, but it reduces pain. It's really not an analgesic, it's a hypoalgesic. It reduces it. But it is important to distinguish between analgesics like opiates, and I see the term is used right below, and anesthetics, without consciousness, without sensation. Actually, without sensation, because you can have a local anesthetic where you are conscious, but that part of your body is not receiving any kind of sensory information. Opium poppies are very interesting. There's only one type that produces what we call opiates. And there it is, a family of drugs known as the opiates. Yes, indeed. And then slip right into calling them opioids. And I think they use the term, slicing the poppy open. That phrase is used right above the figure when the capsules are sliced open. They're sliced. I wouldn't say they're sliced open. The slices, it's a bit like tapping maple trees for maple syrup. The pods are sliced, and then they weep the opium, which can then be scraped off and collected. And boy, this has been around for a long time. And there's 1500 before Christian era Egyptians. They were, and they use the term opiates, medicinal value. Um, I forgot about the warmth. That when you do take a good dose of an opiate, you get this glow, and very often with a pretty good hit with an opiate, you'll go to sleep. You nod off. Occasionally, you'll vomit as you go, which is another story, and that's discussed in here in a little bit. But you do not get aggressive and threaten people with guns and go steal things and rape and pillage. You go to sleep. It's not very socially disruptive unless you fall asleep in the middle of the street. When it was first introduced to the West, people got pretty excited. Laudanum, something to be praised, was used quite a bit. I remember old westerns; they would sometimes talk about using laudanum. So that's nice, and Mrs. Winslow's soothing syrup, a pennyworth of peace, secret liquor cure. So it was sold by our friends at Sears. Now maybe Sears is not part of your life, but it certainly was part of mine. And so this was pretty mainstream. and it was given to children quite a bit. It does quiet them down, and there we go. The laudanum and paregoric and mail order from our friends at Sears. And there's the Harrison Act, which we've seen before, and there's structural uh, demonstrations of the structure of opiates here. The It was the first time the active ingredient of any medicinal plant was isolated, so that was quite a deal in the early 1800s. And I think we can. So what do we have over here? Well, it just talks about how to make things, and heroin was produced by our friends at the Bayer Company, who thought it would be better than morphine, relieving pain without the danger of developing opiate use disorder. No such luck there, but it is important to remember that heroin is converted to morphine in the brain. So by the time it gets to your receptors in the brain, heroin is not heroin, it is morphine. And I mentioned in an earlier lecture that Europe allows heroin. All right, in terms of scheduled drugs, Europe does allow the use of heroin for medicinal purposes when you don't have much person left and they're suffering and you're trying to get morphine into them. Heroin can be concentrated and it goes through the blood-brain barrier really well. So they have decided it's safe. We have not. There are some structures. Let's just look at that for a second. Morphine, interesting side structure there. Codeine looks pretty much the same. Heroin looks remarkably the same. Ooh, naloxone, Narcan. I've done more research in my life with naloxone than any other drug. I'm a huge user of naloxone from before they sold it when it was an experimental drug. So you can see the structures are similar, and you can understand why naloxone would potentially go into the same receptor. And respiratory depression is a problem with certainly with overdoses of morphine. That's what that and cardiovascular collapse. That's what can kill you. With chronic opiate use, constipation can become a real problem. And hi, my name is Tim. I had my hip replaced, I don't know, 7, 8 years ago. And opiates were part of my life for a while after that. And geez, they were obsessed with constipation. I can't tell you how much they worried about constipation. Uh, didn't happen to me, but with chronic use, it can be a real problem. And more drugs, more drugs. There's a very nice table. Certainly no reason to memorize it, but there's oxy in there, and the fentanyl is down there with the totally synthetic narcotics, and that is a very dangerous opiate, extremely potent, and because it's cheap to make, it's one of the reasons where a lot of people have been dying. And there's endogenous neuropeptides, which is what I would call opioids, enkephalins, endorphins, dynorphins, endomorphins, nociceptin slash orphanin FQ, which, from my perspective, my time in the field, that's pretty new. It is very interesting. Bioavailability, blood-brain barrier, most important effects on the CS and the GI tract, below constipation. And there's naloxone, opiate overdose deaths, and naloxone can be very important for this. And there's the term Narcan. And I wish I could find the video I made years ago of um, giving naloxone to a rat that I had given a very substantial dose of morphine to. He was catatonic. You could shape him and mold him. And I gave naloxone in arguably the least rapid way possible. I put it under the skin. No IV, no IP, under the skin, which is a very slow way to administer a drug. And that rat was up and around in under 30 seconds. Naloxone is a very uh, active drug, and it's a very, very safe drug. The I forget whether I told you this story, but physicians used to allegedly pull pranks on junior physicians who were beginning to work in emergency rooms. Someone would come in with an opiate overdose, they'd be on death's door, cardiovascular collapse, and the physician would say, we need to give this person Narcan, naloxone, IV, and once you get the line in, it has to be a slow push. And a slow push means you push it slowly. And of course, it's a trick, particularly if the overdose victim is a female, to get an IV line in someone who's undergoing cardiovascular collapse. And it's probably not necessary, but you get the IV line in, or the needle in the vein, and then you do a slow push, and the physicians who know what's going on step back, and the poor young physician does the slow push, and in maybe 30 seconds to a minute, the naloxone hits the opiate receptors of an addict, and they were feeling no pain, and they were very happy, and now they're feeling pain, and you have a needle in their arm, and they're unhappy because you have probably triggered withdrawal in them, and they sometimes hit you, and the older physicians laugh, and it breaks up the monotony, if there is such a thing, in the emergency room. Um, Now, of course, they distribute this to people, and you can have injectors, but the oral, the nasal is desirable. I forget. I think I have a tab open here. Spoiler alert. Is that spoiler alert? Spoiler. No. Where's my link? Here's Pert. Oh, sorry. This is a. All right. I'm going to get you a picture of the Narcan thing. I'm really sorry it was up. So there's the Narcan nasal spray. Let's just get an image. And they were giving these away in the state of Pennsylvania. And this is the good stuff. You can tell that there's some high tech there, therefore it's going to cost you some money. 4 milligrams is not very much, but remember receptor binding and affinity, and naloxone has really, really high affinity for opiate receptors. And you're dying of heroin overdose, which has turned into morphine. But morphine is going on your receptors and off your receptors and on your receptors and off your receptors, and you're dying. But now we throw Narcan in there, and it just pushes its way to the front of the line. And it goes on the receptor and off the receptor. But morphine doesn't have a chance against it. It is really, really a magical drug. The other tab I have open here somewhere is from Kelly Ross, who makes it. And it's a whole instructional video on, obviously they make a lot of money at this, and it's a discussion of how to use their drug clinically, and it's important and it's good, and they deserve money for this. There is no doubt. There is no doubt. You can, if you saw this drug work once, you would not believe it, uh, how effective it is, naloxone. So there's a discussion of that, and um, this box is good, and it goes on to the next page. And however, naloxone can precipitate an immediate and severe withdrawal. So the victim may be angry and agitated, and that's why the experienced docs are stepping back and hoping that the young doc will get smacked. Um, so, and they're talking about cost. When I first started using naloxone for research, geez, when was that? I don't even want to know. It's like 35, 40 years ago. They used to give us like small peanut butter container jars of the stuff and just say, "Oh yeah, here, see what you can do with this." And now uh, they're getting their money's worth out of it. Naloxone in 2005, $1 per milliliter vial. 2014 is 15. The nasal spray, $125 for a 2-pack, and then oh my geez, $37.50 for injectors. Jeez, it's incredible. Um, some people have trouble taking opiates um, for pain because here's news: all people are not the same, and opiate receptors are all over the brain, and they do different things in different parts of the brain, and there are different receptors. It's complicated, and some people, and I know several, who they vomit so much from taking opiates that they basically can't. And one reason for that is highlighted here. It's the area postrema, which I've mentioned earlier, much earlier. You vomit with that, and it has a weak blood-brain barrier, which is not so relevant to this discussion. It's monitoring your blood for toxins, and it's loaded with opioids, or yes, opioids, because it's your brain's own nervous neurotransmitter. And for some reason, the some people when they take an opiate, that area is affected, and there's vomiting. Um, and there's a discussion of constipation. Diarrhea. I have a tab up for that. Which of these is it? Here. So Imodium, and it's generic now. When you look at it, the structure, I don't know if it screams that it's an opiate, but it's pretty uh, cute the way they've made this. This can influence your GI tracts, and one of the side effects of taking an opiate is you get constipated. And if you have diarrhea that's annoying or threatening you, you would like to be constipated. You would like your GI tract to calm down. And this drug can get to your GI tract and stop motility and stop diarrhea, but it is structured so that if you have a normal blood brain barrier, it doesn't get into your brain. Although some addicts are doing odd things with it, ruining it. I think they're now starting to. You used to be able to buy a little pill bottle. These things, they're tiny pills, and you could get 50 or 100, and they'd be dirt cheap. But now, because addicts are starting to, take a pound of these just to get some of it into their brain and get an effect out of it, they're starting to not market it that way anymore. They're making these blister packs and fewer pills in the container, and of course they want to make money, so they charge a fair amount of money. And for those of us who use it for diarrhea, it's less convenient and more expensive, but I guess it's good for society. So why am I complaining? Um, Boy, those are some beautiful, beautiful data there. And we've talked about receptor binding and Bmax, and there's the binding of tritiated H3 naloxone, and that lets you know how many receptors there are in there. So we can use this technology if we're studying different kinds of people or rats or people who've been addicted or are addicted, and it's very useful. And this figure here is very impressive. Correlations. Correlations can be positive or negative. No correlation means you have a correlation of zero. No correlation looks like you took a shotgun and shot it at a wall and you just have a blur of pellets. There's no pattern. This is as close to a perfect positive correlation as you get in behavioral or biological sciences. So we're measuring two things: inhibition of electrically induced contraction of the intestine, uh, and we know that opioids are involved in that, but these intestines have been taken out, uh, the guinea pig ileum, and then binding, and there's binding on the right. So you have how much does the drug inhibit contraction, which is a behavior, and on the left you're looking at KD, and oh my goodness, what a correlation. It's amazing. It's not a perfect correlation, but that correlation has to be a perfect correlation, can be one of two things: plus 1.0 or minus 1.0. A perfect positive correlation, which this approach is, or a perfect negative correlation. Um, Both of them are equally informative. What you want in science is knowledge, prediction, power, and either approaching positive one or negative one buys you that. And this is probably somewhere like 0.95. It's just incredible. And there's a lot of drugs in there that are discussed in the rest of the chapter. And there's something I saw. So there's Candace Pert and Solomon Snyder. Pert was Snyder's graduate student. She was, it was her dissertation research under his tutelage, where she was looking for opiate receptors in the brain. And as the story goes, they had ground up brains in vials going into a scintillation counter, which measures radioactivity. And they did things in triplicates because that's what you do. And there's all these little vials, and they move in front of a radioactivity detector, and they put the stuff in the machine, and they went to lunch or dinner or something, and they came back, and they looked at the printout, and I think they went out for a bottle of champagne because they could see from what was coming out of the scintillation counter that they had discovered that there were opiate. Well, originally we called them opiate receptors because there were no opioids, but they had found opioid receptors in the brain, and they act just as you would predict. Unfortunately, Snyder and Pert fought a little bit about who owned primacy to that discovery. Let me find her. There she is. So there's Candace, um, sadly dead. She was a force of nature. I was lucky to meet her a few times. Really, really amazing mind. And there she is as a graduate student. And there is the ever thoughtful Solomon Snyder, who apparently plays an amazing classical guitar. And that was probably taken at about the time that they discovered the receptors. And there's another good. Whoops. There's she went into business after this. There we are. No eye protectors, I noticed. Doing some pipetting. Life was simpler then. Not all lab, I don't think that would meet lab protocols of the modern era, but they were being considered for the Nobel. Never quite happened, but let's look at Solomon Snyder for a second. When I first came to Scranton, I used one of his books for a course I taught. He wrote really well. He's not dead. So there he is. He's moved to bow ties, and wide-ranging contributions. He's still alive, and he won. I thought it was here. He won a Lasker Award, which is sometimes thought to be a stepping stone to the Nobel. But they are both uh, top shelf intellects. All right, she's dead. She's dead. Let her go. But they're both really impressive, and they'll come up again, and their research will come up again. And there's affinity. Remember, you might look back at receptor binding and receptor kinetics, but you have receptor sites and you have ligands, and ligands are not all created equal, just like people are not all the same, and they have different abilities to bind. And there are receptor subtypes, and the first three here, mu, delta, and kappa, I think about a fair amount. Mu has been the majority of my life because that's the primary but not exclusive pain inhibitory receptor, and it's the primary target of naloxone, but not exclusively. And there's mu and talking human brain postmortem. Looking at that, it's can you tell me how that brain is cut? How has it been sectioned? And the answer is that's horizontal. And can you tell me which way the animal is aimed? And that's pretty easy because you can see the olfactory bulbs on the right side. So it's aimed to the right. Notice that, by the way, in us, we don't have much olfactory bulb, but boy rats do, and they have opioid receptors. And the cerebral cortex is here. And look at all the receptors in the frontal part of the cerebral cortex. And one thing that opiates are very good at relieving is suffering. And the frontal cortex is involved in that. And boy, if you care about people and you want them to die with dignity, you would like to alleviate suffering. Unfortunately, opiates are not very good for long-term pain. There's the striatum, Kapoor. This part of the cerebral cortex does have some binding, but not as much as up here. Here's our friend, the hippocampus, which looks all curled up, and the dentate. We've seen that before, CA1. We've seen that, and as soon as you see dentate and CA1, you start sweating. Oh my God, we're going to start talking about NMDA receptors and LTP, aren't we? I think not in this chapter, but if you got a little panicky, that's good. Thalamus. Thalamus is the great sensory relay station of the brain through which all sensory information on its way to the cerebral cortex relays. That means neurons come in, talk to other neurons, and then the second neurons project to the cerebral cortex. Boy, there's a lot of opioid receptors there. And the midbrain is important to me because the PAG is important to me. And it says PAG here, but it doesn't label it. And it also says Raphe nuclei. Dorsal and median Raphe have a lot. So structures that we've been talking about all along are coming up again, hopefully helping you feel more confident about prepping for the comprehensive final, because information is used over and over and over again. And there's a list of structures again. Median Raphe is specifically mentioned. Spinal cord is there. Some effects of opiates can inhibit pain in the spinal cord directly. Other effects of opiates inhibit pain by working in the brain. And I've already given you my lecture on dorsal and ventral non-opioid and opioid descending brain to spinal cord. And Raphe Magnus comes up in this chapter. So we've already sort of gone over that. So hopefully that helps. And there's respiratory depression, which you do not want, and cough control, which sometimes you do, nausea and vomiting, brainstem, that's area of postrema. So it's big. It's everywhere. Oh look, substantia nigra, nucleus accumbens. Hello. So certainly with accumbens, and we'll see there's also VTA where we should be thinking about reward and addiction. And then there's different subtypes, delta on this page. And species differences. Yes, that's true too. And here's, what do we got? Looks like rats again. The plane of sections, the same. These are horizontal sections like the other page, though the other page was colorized, where the density of the receptors, the autoradiograms, the silver grains were turned into color to make it easier for you to see. Here you're looking at the silver grains. And if you think back to something I'm kind of obsessed with, what kind of microscope are we using here? How are we imaging this? The field looks kind of what? Dark. So this is dark field again. And these are autoradiograms, and those are silver grains. And silver grains are little mirrors. And when the light, where there is no silver grain, is going through the tissue, it just goes out and doesn't hit the objective. But where there are silver grains, it gets bounced around, and a bunch of it goes, or a percentage of it goes into the optics of the scope, and you can see it. So Kapoor, caudate there's a lot in the thalamus. There's Raphe. It says nuclei, dorsal and median both have some. Locus ceruleus, which should make you think of what color, and should make you think about what neurotransmitter. The color is blue, cerulean blue. The neurotransmitter is norepinephrine. And norepinephrine rears its head in this chapter a few times. And there, hello, is the all curled up hippocampus. And there's the striatum. Again, notice the pattern is very different for delta receptors. Notice the olfactory bulbs are really lit up. Cortex has a somewhat different pattern to it. Not so much in Raphe areas. And then you get kappa and olfactory bulbs, and the cortex doesn't seem all that important. You still get some striatum, some thalamus. Raphe lights up again. Locus ceruleus lights up. So, it's complicated. And you thank your lucky stars that it's open book. Hallucinations. Kappa. There was something that grows wild locally, a form of salvia that up until maybe 8 years ago was legal. And salvia is an extremely selective kappa receptor agonist. And that can produce pretty intense hallucinations for seconds. Pain, gut, dysphoria, not euphoria. NOPR, analgesia, feeding, learning, motor, neuroendocrine. And then we're cloning things, and there's a discussion of how to clone things, and they're linked to G proteins, and we've seen G proteins before, which means They're not going to be very fast, but their effects can be fairly profound. And there's one that's been characterized, and there's a classic uh, metabotropic receptor and those are the transmembrane elements of it, and that's delta. And be amazed that you live in a world that there are people somewhere smart enough or well trained enough to figure that stuff out. Um, they didn't mention one of my favorite people in terms of discovery, little Hans Kosterlitz. Um, but there's a nice discovery there of endorphins and how they're synthesized and early on in terms of neurotransmitters. Peptide neurotransmitters were being mentioned as being unusual, and they have large propeptides, and the Golgi apparatus comes into play, and things are packaged and shipped. And as they're being shipped, the peptides are being cleaved, and subunits are being cut out, and those are the effective neurotransmitters. And we can tell where things are if we have clever enough technology by looking at their messenger RNA, DNA, RNA, protein synthesis. Um, this this is impressive. I can't add to it. I do like it quite a bit. When I used to lecture about opiates, because I was still a little mad when we had discovered dorsal and ventral non-opioid and opioid, that people were insisting on being obsessed with opioid. And I've said drug companies have been modifying chemicals for decades to try to make a non-addictive uh, opioid or opiate analgesic. I said we should be focusing on the other one because that one doesn't show tolerance or cross tolerance and probably won't show addiction. But I'll tell you, as we're getting more molecular and we understand these things in intimate detail, we may be getting to the point where we may be able to develop things that have analgesic potential with reduced side effects, like addiction. Now, I wanted to draw attention here. Remember back a couple chapters, we were talking about behavioral assessments for pain, and I did most of my research with the tail flick, uh, which is a spinally mediated reflex. But people also use hot plate, where you put an animal on a hot plate, and that's the term. And that involves brain processing. So hot plate is used here. And that's a nice discussion, and it brings in behavioral tests. Things are being used and reused. There's hot plate again. And it says the hot plate test reached a maximum response of 87% compared to morphine's maximum of 92, which is pretty similar, and meaning that this newly developed ligand works pretty darn well. It does seem to use opioid receptors, and this is cute. The finding that PZM21 did not activate dopaminergic reward pathway nor induce conditioned place preference. And remember that, where you associate a location with a drug to see if the animal goes back to it later, indicates that the molecule is not likely to cause typical opioid reinforcement or opiate reinforcement or abuse. I stand corrected. The people who do this research are beginning to close in, I think, on analgesics made for opioid systems that may be a lot safer. I was wrong. So there's the propeptides. Uh, I I was almost going to subject you to this, but I'll try to suppress. When I wasn't doing brain stimulation, I was producing what we called stress-induced analgesia. And just like the brain stimulation, We had an opioid type. It actually turned out to be two, and those two could be blocked by naloxone, indicating that mu-opioid receptors were probably involved, and a non-opioid, which we never fully characterized, but almost certainly was endocannabinoid. And there were stressors there. Foot shock is what we used. We didn't do restraint, swim stress. They were starting to do that as I was leaving. That was pretty stressful. And there's a discussion of that and analgesia partially blocked by naloxone. It depends on the type of analgesia. Some forms can be tremendously reduced by naloxone, and others just aren't touched. No doubt because there are multiple systems, just like with the brain stimulation. And then there's co-localization, and what are they localized with? And things we've talked about in previous chapters. And pro-hormones distributed. Yep. Hopefully these chapters are in. We've seen this figure before. And so those are neuroendocrine cell bodies in the hypothalamus producing corticotropin releasing factor onto the blood vessels in the stalk of the pituitary, and magic ensues, which can influence the anterior pituitary. And there's ACTH, and we've dealt with that, and beta endorphin acting as a hormone, like the ACTH. And we did research with the pituitary removed from animals, and some one form of opioid stress-induced analgesia. You take out the pituitary, it doesn't happen. So that form of stress-induced analgesia involves this circuit where it is releasing ACTH and beta endorphin into your general circulation as if you got an injection of a drug. At that time, did I mention, because whenever I think about addiction in this chapter, it deals with it, I also think about love, that people, mostly female people, get pituitary tumors. And now we destroy that tissue, often with radiation, gamma rays, and we just take the pituitary out. And that women who've had their pituitaries destroyed tend to get married less often than people with pituitaries. And when they do marry people, they tend to, less so than people with pituitaries, report that they're marrying for passion. They are marrying for companionship. So love is an addiction, and that gland and possibly those very cells are involved in addicting you passionately to people, I think, although I haven't looked at that literature in 20 some years. So there's subtypes, and I under functions, notice that the NOPR, spinal and analgesia, supraspinal, pro-nociception. Really interesting. That's a complicated one. So that table is of interest. Okay, the cellular changes. So let's look at this one, and we're going to be revisiting lots of things we've seen before. So there's postsynaptic inhibition opening potassium channels. So there's your G protein metabotropic receptor, which is affecting a potassium channel. And hopefully you remember that at rest, the resting potential is not the equilibrium potential for potassium. If you open extra potassium channels at the resting potential, potassium is going to leave that cell, and positive ions leaving a membrane system will make it more negative. It will hyperpolarize the membrane, which will move the membrane away from the threshold of activation and inhibit the cell. So there we go. And then we have axo-axonic connection closing calcium channels. So there's a metabotropic receptor again, and a G protein, and why are we doing that? It's on the axon terminal. Well, remember how neurotransmitters are released, and calcium influx through channels. Calcium flows into the cell if those channels open, and if you inhibit that, you're going to reduce the amount of neurotransmitter that's released from that terminal. And so both of those are going to reduce transmission. And then there's presynaptic autoreceptors. Largely the same effect. So opioids calm things down. Cyclic AMP cascades, we saw that with second messengers, and also with NMDA receptors and memory. And the cascade has been implicated in chronic effects of opioids, including tolerance, dependence, and withdrawal. And we'll see those again. And pain. Okay, so here they choose to teach you some of the basic neuroanatomy, neurophysiology of pain. And the way I like to do this is to imagine you're in your home or dorm and you go to fill up the tub or a sink and you turn on only hot water and you forget. And you plunge your hand into scalding water. Your hand is going to come out of that water before you feel any pain. That's a spinal reflex. That's like the tail flick. But now you're going to feel two pains. There's going to be a first pain. Why first? Because it's first. You get a first pain that when you describe it, it feels bright and sharp and pricking. And then you get a little bit of quiet. And personally, I curse in this interval. And cursing has its own neural circuitry. And the PAG is probably involved. And if you want people to curse, hurt them. People who can't speak voluntarily, you hurt them. They can sometimes curse a blue streak, but that's another point. And then after this quiet time, you get second pain because it arises second. And second pain is dull, throbbing, and aching. And it is a more emotionally laden. Now, most of our research on animal pain admittedly deals with first. Most of human suffering probably comes from second. Let me see what they do on the next page because I do have a little supplement here. Okay, my supplement. Pain can kill. Oh, I have to come back to that. Let me close some windows. No, I'll need them if I don't close them. Okay, neuroscience for kids. So if you want to find this, I can show you. I searched for Chudler, which I guess you'll just have to remember. He's Eric Chudler. By my eye, he looks a lot like Bill Nye the Science Guy, and he's in Portland. No, he's in Seattle. And when I was at UCLA, he was my undergraduate research human. And now he's all grown up and he's doing great things. So I searched for Chudler conduction velocity, and he runs this website, Neuroscience for Kids. And while we're on that, now I'll come back to it. And you can look at it and say, oh my, this doesn't look very modern. This looks like a lot of my websites did in the 90s, but it's pretty brilliant. And so there are primary afferent, those are your sensory axons. And I talked about, wrapping myelin around axon, a little bit like wrapping things around hot dogs. And you can see that you have three myelinated axons there. And remember, myelin is put on axons. It's not part of axons. And in the peripheral nervous system, hopefully you can name the cells that do that. And they have to be capitalized because it's a human name, Schwann. And moving an action potential through an axon is a lot like moving water through a pipe or trying to breathe through a straw. And if you want to breathe through a straw, pick a fat straw. So these axons are faster, and then these are a bit slower, and these are a bit slower. And oh my, over here are fibers with no myelin. And if you find a primary afferent with no myelin, you can bet the rent that it's a C fiber. And C fibers are important to pain, as are the smallest of the myelinated, the A deltas. Now, below here, you have speed, meters per second. And for A deltas, 5 to 35, and for poor C fibers, 0.5 meters per second to 2. That's slow. And there's some nice pictures of myelin, and here graphically it looks very fat, but the laminations are very tight. And then here's a graphic. I don't know where he found that, but this is pretty cool. So here is speed in miles per hour, and over on the far right you have an airplane, but right next to that you have A beta nerve fiber. They're fast, over 200 miles an hour. And when a physician does a knee jerk test on you and whaps your knee with a little hammer, those puppies are involved in that one. But not pain. Pain is way over here. And way, way, way over here are C fibers. How slow are they, Johnny? You could outwalk them. You can walk faster than action potentials propagate along a C fiber. They are slow, and that's why they produce second pain, the slower pain. Where's the first pain? That's over here, A delta. And that's a lot faster than walking. It's somewhere between an Olympic sprinter and a fast bicycle. So it's, I guess it's closer to an Olympic sprinter, but it's fast. And that's first pain. So again, first pain, bright, sharp pricking. Second pain, dull throbbing and aching. And you probably are not going to be surprised to find that they have different pathways and different functions in the brain. And there's a discussion of early and late. I say first and second, they say early and late. It's the same. And then there are pathways. And there's your A deltas and C's. Now, one thing that's a lie here is that in this picture, you'll see that the synapses onto the second cell bodies, the primary afferents, come in from your skin, and then they talk to second cells, which in this picture are outside the gray matter of the dorsal of the spinal cord. They're out in the myelin. That's not true. Both of these synapses are going to be in the dorsal horn. Yeah, we can live with that. But each of them synapse in the spinal cord. And then the axons from the second cell cross and ascend to the brain. And one thing I'd like to point out is, there's a pathway, there's thalamus, there's the cerebral cortex, that's all important. Oh, look, periaqueduct gray. And it is getting axons in this picture only from the red zone. And the red zone is the slower C fibers, and that's the one that produces emotions. And I believe those are the ones. In there is a pathway that can trigger profanity, but that's just me. And more talk about first and second, and Then we're looking at, I think these are MEGs, yes, MEG traces, and you can look back at what MEGs are. And there's early pain, and it looks like just regular sensory stuff. And then you start getting second or late pain, and you get secondary somatosensory cortex, and then anterior cingulate, which is important for a lot of things. It keeps coming up in the textbook, and it's very important for pain. And the here's a sagittal section. It very nicely shows you um, the anterior cingulate, along with somatosensory type areas. There's PAG, so that's two significant descending pathways originating in the PAG. How, thank you for mentioning that. A reference would have been nice, but that's the research I pointed out to you earlier. Oh no, they're not talking about me. They're talking about two opioid pathways, and they're not wrong. They're not wrong, but there's also non-opioid, which they're just skipping. And we're going to have a graph here in a second to show you, yes, how things can happen. So here are your primary afferents, and free nerve endings are most important for receiving pain information. Why are they called free nerve endings? Because they're nerve endings that just branch freely in your skin. And there's your sensory afferent. This is in your dorsal ganglion between your vertebrae. One branch goes out to your skin, the other branch goes into your dorsal horn, and it excites a projection neuron. The second neuron is what crosses and goes to the brain. And here's a descending pathway that can directly inhibit it. Here's a descending pathway that can inhibit an excitatory interneuron. So in other words, here the pain information would go from the primary afferent to an interneuron, and from the interneuron to the projection neuron. And here we're inhibiting the inhibitory interneuron or excitatory interneuron. And then finally, which I particularly like, you have an opioid neuron. Therefore, that must mean there are opioid receptors over here. So therefore, if you put morphine directly in here, they would work here. But here you have descending pathway that excites the opioid interneuron. Then the opioid interneuron, once it's excited, inhibits the projection neuron. And if you inhibit the projection neuron, even though you're trying to excite it, if you inhibit it, you block pain. So that's way oversimplified. I usually spend a week on this stuff, but maybe a little bit more. And GABA is there. And all this should seem more and more familiar. There's a discussion of accumbens, amygdala, thalamus, sensory pain scores, whereas affective pain scores, anterior cingulate, thalamus, and accumbens. So the sensation of pain and the motivation produced by pain are being processed differently. It's and there's, maybe I've skipped over it already, but there's a mention in here about opiates where the person getting the opiate, you ask them, how's it feel? And they say, oh, the pain is still really strong, but I don't care. The suffering is gone. Not necessarily the ability to perceive what kind of sensory information is coming in. And electroacupuncture is discussed more than I'm used to in this chapter, and different frequencies. High frequency, in this case, 100 hertz, HERTZ. It's a human, so it's capitalized. It just means cycles per second. So 100 cycles per second, 100 pulses per second. And then the slow is 2 hertz, and they're different. and they have different effects in the central nervous system. And there's free nerve endings, and so that's, I think they have the synapse in the right place here. Oh, Ref-A, look, descending PAG to Ref-A, and Ref-A magnus would be most important here. So we like that. And there's PET scans and sustained pain, and there's nucleus accumbens and anterior cingulate. And then we get into some pretty real, some chemistry and gene therapy, maybe. And we're getting the ability to do some pretty amazing stuff. And then dual mu and NLPR agonists, where the drug is sloppy because it does two things at once, but by that it can possibly have therapeutic effects without some of the negative side effects. This pharmacological approach and the structure-based drug development described in the box are promising advances in the search for safer painkillers, I hope. Okay. Opioid dependence. There's a term in the middle of this, high intensity opioid prescribing. I told you that if you want something to be addictive, you want it to hit you hard. And high intensity is a code name for hit you hard. Now, sometimes you have to do that for ethical purposes, but when you're doing that, you should know you're doing it because the patient has had an experience that can lead to addiction, as opposed to a low intensity experience. And I recommend opiates to effect. You don't have fun with them, you just make it so you're not suffering. And there's risk factors. There's, one second. There's SNP analysis. Again, this is all stuff we've seen in other chapters. There's a meta-analysis again. And this is an important message for medicine. Meta-analysis suggests that opiates are much less effective for chronic non-cancer pain than some other methods. If you're suffering right now because you've had a hip replaced or your arm is broken, geez, take it. But chronic back pain, no, it's not good for chronic. Okay, they're talking about how to minimize what they call the opioid problem. Let me show you my advisor, now sadly dead. So John C. Libeskind died at 62 thanks to cigarettes. You use them correctly and they kill you. And he wrote an editorial for the journal Pain, and the title, where's the title? Pain Can Kill. And he was important in pushing for adequate pain relief in hospitals in the early 90s. And why he did that was a doctoral student in nursing came into his lab and told him horror stories about recovery rooms being like Dante's Inferno because of all the suffering that was being experienced because they weren't adequately medicating for pain. And she wanted to see if that had health consequences other than ethical consequences. And she showed that pain experience can suppress your immune system. And if you're in there for surgery to remove something cancerous, cells have been kicked loose by that surgery. And if you now experience uncontrolled pain because they let you, your immune system is suppressed, and those cancer cells are more likely to migrate and set up again. Uh, Pain can kill. So we're in a balancing act now. There is an opiate epidemic. It was largely fed by physicians physicians being misled and giving out too many opiates. But people in emergency rooms, people who've had their hips replaced, they need medication. You don't medicate them adequately, you're increasing the likelihood that they'll get a post-surgery infection. So we have to walk an intelligent line here. Uh, Pain can kill. John C. Liebeskind. Love you, man. Um, okay. And there's meta-analysis again and medical boards. Boy, I go to meetings for this and I speak out for pain sufferers, and the tide has turned. Everybody is blaming trying to have patients being comfortable for this pain epidemic. No, it's look at the number of pills that the pharmaceutical industry was distributing, and the lies about oxy and just okay. So potassium chloride, all the same stuff, and dopamine receptors. Dopamine receptor antagonists are seen to block the reinforcing effects of opioids. Yep, dopamine rears its head. Addiction rears its head. And hello, VTA, nucleus accumbens. We haven't talked about the pallidum much, but there we go. Things we've all seen before. Now here's a word. Oh, there's 6-OHDA. Now this was mentioned earlier. 6-OHDA reduces but does not abolish the reinforcing value of heroin, suggesting that dopamine is not the whole story. Salience. Remember my friend who wrote the book on nicotine not being addictive? He talked about salience, and my former students who went into addiction research and simulated the VTA and did microdialysis in the nucleus accumbens, core and shell, they talked about dopamine and salience. And what that means is your brain is telling you, whatever's going on right now, this is important. Pay attention to this. And that can happen with smoking. And it's part of addiction process, other addiction processes too. So it's important. It's different than reward, but it's very important. And there's tolerance and cross tolerance and sensitization, physical dependence, and acute effects of opioids, really opiates and withdrawal symptoms. And boy, look at the heroin versus methadone. And as I've said and said again, you want something to be addictive, hit them hard. And boy, heroin hits you hard. But methadone is much kinder and gentler. And there's a nice discussion of methadone. And there's oxy again. You can smash it. It's time release. It was supposed to be something like not quite methadone, but more like that, slow, gentle, therefore not so addicting. But people found that if you smash the heck out of it, you could break the slow release capsules and suddenly it would come out. In my mind, I would have added naloxone in capsules so that if you smashed to release the oxy, you would also release the naloxone, rendering it neutral, which now they sort of are doing for other drugs. And doctor shopping, there's a famous conservative talk show host. He may be up to 3 marriages. He doctor shopped to feed his addiction. Boy, look at fentanyl. Jeez. I mean, heroin is already quite potent, more potent than morphine and the fentanyl. You make a base 10 error while diluting things with fentanyl in it, and trouble ensues. And here's a map of opioid deaths, which is in your textbook. If you blow it up, there we are, and there's Luzerne County. So we've got stuff going on here. Plus we have lead. So there's theories of addiction and model of tolerance and cyclic AMP, and that's a very nice figure, and it is supported to some extent, and not some. There's good data to support this, and some of it is in people, and some of it's in cell culture, where the tissues are living in dishes. I don't know why I'm a fan of downregulation, and therefore I'm obliged to say, hence, downregulation is not a likely explanation for morphine tolerance. Okay, wrong again. And here comes classical conditioning, and Siegel is very important in this, and you deserve that lecture, but I don't think you'll ever get it. And triggers, classical conditioning, again. Under what circumstances individuals develop drug enhancing associations or drug opposing responses is not clear, but the mesolimbic dopamine pathways may be involved. Good guess. And now we're moving into treatment programs, and it's complicated, and detox and maintenance of some drugs can be very helpful. And there's one of the methadone. And there's, again, electroacupuncture and the high frequency. Methadone maintenance, there are those that are like morally, religiously opposed to this, which means they're hurting people. And they're probably being supported by people who sell opiates illegally on the streets, because this works. And it works. And methadone treatment programs save taxpayers, if all you care about is money, $12 for every dollar spent. By the way, that's about the ratio for infrastructure spending. $10 saved for every dollar spent. I just had a friend whose front wheel was ripped off his car by a pothole in Scranton. Completely ripped off, like $800 of labor by a friend to put it back on. Infrastructure, the little people pay, not the big people. Buprenorphine, it definitely has advantages over methadone, and there's fewer clinics, so people who live near the clinic are less disturbed, and they have a right to be disturbed. The implantable option, that's like magical. Under the skin, you can't abuse it. I'm sure cost is an issue, but boy, that is elegant. Pregnancy, geez. You know, addictive drugs and pregnancy don't go together. The book ends up being fairly positive about buprenorphine and pregnancy. And there's meta-analysis. Meta-analysis suggests there's no difference in efficacy associated with methadone compared to buprenorphine, but buprenorphine is easier. Antagonists, naltrexone, for example, but the person taking it is an important variable. Unfortunately, this method appeals to only 10% of the addicted population. But boy, when it's right for you, it's right for you. It works pretty well. Counseling, sure, multidimensional, why not? add it. But the other drugs are definitely worth adding to just talking. Your brain has changed, and you have to deal with that. Well, okay, so we have done the opiates. I ran over like I always do, but you can always hit pause. I don't like to.